Pacemaker current. The pacemaker current, or FORIC, also referred to as the funny current, is an electric current in the heart that flows through the HCN channel or pacemaker channel. Such channels are important parts of the electrical conduction system of the heart and form a component of the natural pacemaker, first described in the late 1970s in Purkinje fibers and sinoatrial myocytes. The cardiac pacemaker funny if current has been extensively characterized, and its role in cardiac pacemaking has been investigated. Among the unusual features which justified the name funny are mixed Na plus and K plus permeability, activation on hyperpolarization, and very slow kinetics. Function. The funny current is highly expressed in spontaneously active cardiac regions, such as the sinoatrial node SAN, the natural pacemaker region, the atrioventricular node AVN, and the Purkinje fibers of conduction tissue. The funny current is a mixed sodium-potassium current that activates upon hyperpolarization at voltages in the diastolic range, normally from 60/70 MV to 40 MV. When, at the end of a sinoatrial action potential, the membrane repolarizes below the if threshold, about 40/50 MV, the funny current is activated and supplies inward current, which is responsible for starting the diastolic depolarization phase DD. By this mechanism, the funny current controls the rate of spontaneous activity of sinoatrial. Another unusual feature of if is its dual activation by voltage and by cyclic nucleotides. Cyclic adenosine monophosphate CAMP molecules bind directly to F channels and increase their open probability. CAMP dependence is a particularly relevant physiological property, since it underlies the if-dependent autonomic regulation of heart rate. Sympathetic stimulation raises the level of CAMP molecules which bind to F channels and shift the F activation range to more positive voltages. This mechanism leads to an increase of the current at diastolic voltages and therefore to an increase of the steepness of DD and heart rate acceleration. Parasympathetic stimulation, which acts to increase probability of potassium channels opening but decreases the probability of calcium channel opening, decreases the heart rate by the opposite action, that is, by shifting the if activation curve towards more negative voltages. When vagally released acetylcholine, H binds to muscarinic M2 receptors, which promotes dissociation of BG subunit complexes, leading to direct opening of the G protein-gated inwardly rectifying K plus channel GERK/Curricatch. Related currents. A similar current, termed hyperpolarization activated, has also been described in different types of neurons where it has a variety of functions, including the contribution to control of rhythmic firing, regulation of neuronal excitability, sensory transduction, synaptic plasticity, and more. Molecular determinants. The molecular determinants of the pacemaker current belong to the HCN channel hyperpolarization activated cyclic nucleotide-gated channel, of which 4 isoforms HC-none to HCN4 are known. Based on their sequence, HCN channels are classified as members of the superfamily of voltage-gated K plus KV and CNG channels. Clinical significance. Because of their relevance to generation of pacemaker activity and modulation of spontaneous frequency, F channels are natural targets of drugs aimed to pharmacologically control heart rate. Several agents called heart rate reducing agents act by specifically inhibiting F channel function. Ivabradine is the most specific and selective if inhibitor and the only member of this family. Recent studies have also indicated that funny channel inhibition can be used to reduce the incidence of coronary artery disease outcomes in a subgroup of patients with heart rate equals 70 bpm. Cardiovascular diseases represent a major cause of worldwide mortality, and the relevance of the genetic component in these diseases has recently become more apparent. Genetic alterations of HCN4 channels, the molecular correlate of sinoatrial F channels coupled to rhythm disturbances, have been reported in humans. For example, an inherited mutation of a highly conserved residue in the CNBD of the HCN4 protein S672 is associated with inherited sinus bradycardia. In vitro studies indicate that the S672R mutation causes a hyperpolarizing shift of the HCN4 channel open probability curve of about 5 MV in heterozygosis, an effect similar to the hyperpolarizing shift caused by parasympathetic stimulation and able to explain a reduction of inward current during diastole and the resulting slower. Biological pacemakers, generally intended as cell substrates able to induce spontaneous activity in silent tissue, represent a potential tool to overcome the limitations of electronic pacemakers. One of the strategies used to generate biological pacemakers involves the use of cells inherently expressing or engineered to express funny channels. Different types of stem cells can be used for this purpose.